The article titled Selective Small Molecule Targeting of Triple Helix Encoded by the Long Non-Coding RNA Metastasis-Associated Lung Adenocarcinoma Transcript 1 or MALAT1, published by Abulwardi et al. In 2019, studies the effect that small molecules have on the triple helix of this MALAT1 and how that affects its stability. Before, the non-coding RNAs were considered non-functional and they were ignored. However, the study by Zhang et al. In 2017 proved that there are many types of cancers that show abnormalities in their non-coding RNAs, specifically long non-coding RNAs. In fact, studies since then proved that the overexpression of MALAT1 can lead to an increase in proliferation and migration of non-small cell lung tumors. They also mentioned that MALAT1 has a triple helix instead of a poly-A tail, which stabilizes it. So Abulwardi et al. In this study hypothesized that MALAT1 is able to stay in the nucleus because of this triple helix stabilization. This is important because being able to pinpoint the location of stability of this MALAT1 allows us a target which we can then cause a destabilization of MALAT1 and decrease migration and proliferation, which is a huge potential treatment for cancers that show this non-coding RNA abnormality. So how does MALAT1 affect gene transcription? The study by Amodio et al. In 2018 showed that MALAT1 can affect gene transcription in 4 different ways: through mRNA splicing regulation by phosphorylating the SR proteins, through trimethylation of histone 3 and lysine 27, by increasing the transcription factor bindings to promoters, and by inhibiting the microRNAs. So Abulwardi et al. Did a small molecule microarray library screening to find compounds that were able to bind to DNA and RNA. They found these 2 compounds, compound 5 and compound 16. They then put a fluorescent tag on the MALAT1 of mammary mice tumors, which have a tendency of migrating to the lungs, and treated them with compound 5 and compound 16. These cells showed a decrease in proliferation compared to this positive control, which is the ASO, also known to decrease the accumulation of MALAT1 in the nucleus. However, it doesn't affect the triple helix, and their negative control, MOCA and DMSO. So compound 5 showed a decrease in proliferation, and so did compound 16, as well as a decrease in branching of the cells. Abulwardi et al. Went on to do a frat analysis of how these compounds were going to bind to the triple helix. So they used the Autodoc tool 4, and this is an algorithm in which an algorithm that tells you where these compounds would most likely bind to the triple helix. And they pinpointed these higher density areas of compound 5 and compound 16. And then they were able to find that compound 5 actually binds to the major grooves of the triple helix through van der Waals interactions, and it causes the triple helix to compress. Compound 16 binds to the minor grooves through hydrogen bonding, and this causes the triple helix to elongate. Both of these destabilize the triple helix. So this study was able to show how these compounds, compound 5 and compound 16, bind specifically to the triple helix of MALAT1 to destabilize it and therefore decrease the concentration of MALAT1 accumulating in the nucleus. This is a huge potential treatment for cancers that present these triple helix motifs in their non-coding RNA abnormalities. For future projects, the compounds can be tested on other non-coding RNAs that don't have the triple helix and present other motifs or other abnormalities.